I am going to briefly give you an overview of some recent developments on our team that our team has been working on and what we have been implementing also on our platform and as training. And I'm sure that Mohit will try and figure out his slides so that he can kind of pick it up from there. So briefly to get started, just 1 second, I need to finalize just one thing. Okay, there we go. Okay, so let me share my screen. I have a few exciting things that I want to share with you today, and those include, I'll start from kind of the major areas where our team is focused. And over the past several years now, we have been working with a variety of partners and research collaborators and really trying to connect education with self-service tools or a platform that we've been building for big data analysis, as well as supporting different research projects with services. And our goal is really to help translate bioinformatics research to applications, and we do so in several different areas. So one of them is oncology, infectious diseases, neuroscience, and biotechnology. And so what we want to share with you today are some of the ways that you can learn about some of these topics as a student, potentially as a researcher or as a faculty member. You can think of this as an opportunity to also take some of the resources that we've developed and use them in your training when you want to supplement your curriculum with some hands-on practical exercises that anyone without any technical background can leverage. But also think of it as an opportunity to leverage the big data that is being collected in all of these domains and apply some of these tools to analyze such data, develop hypotheses, potentially utilize some of these methods in your actual research. So what are some of those directions in these domains? In each one of these domains, we are looking at really an explosion of different types of data, such as genomics, transcriptomics, epigenomics, metagenomics, many other types of data that essentially speak about biological phenomena at very detailed molecular kind of level, right? So as a result of that, there is a challenge to process, analyze, and integrate such data in a meaningful way. And when I say meaningful way, what I really mean is to be able to in an accurate way, identify and differentiate between signal and noise. That's challenge number one. And then challenge number two, connect the dots in this complex puzzle to a biological phenomena or a signal that we can interpret biologically. And so to do that, a variety of methods in all of these different types of data have to be easy to use, have to be accessible. They have to be understood, and that's where the training comes in. But also, in many cases, they still require a lot of time, and that's where our team can help and assist either by mentoring or by actually providing those services. And so I'll talk a little bit about how we are doing that and briefly describe some of the tools that you can even try today. So what primarily I will be talking about today is not the training component. I will leave that for Mohit to add on at the end of my presentation, but I will actually speak about some of the tools that we use ourselves and some of the tools that you can also leverage in those kinds of research domains and how they practically address some of the bigger challenges in each one of these domains. So we've been working with the Tauber Bioinformatics Research Center, a center out of Israel, at the University of Haifa in Israel, that has been developing a comprehensive platform that allows anyone without the technical background, so a biologist or somebody coming from a life science background, to leverage these big data tools to be able to process, analyze, visualize, and integrate a lot of these types of omics data. And so some of the tools that I'll speak about today are commonly used, but I'll speak about specific areas of application where they can make a big difference. But before I jump in those topics, I will briefly give you an overview of what this platform is and how you can use it today, just to understand what this really is. So to kind of learn about the platform and take a look at it, I want you to please follow me on this link, server.t-bio.info. This is a platform that, again, you can register for free. The free registration is going to give you demo access to a lot of the tools that I will be describing today. And you'll be actually able to try some of them in the context of these project examples. Some of the project examples I will also mention. So once you are in this, let me just make this a little bit bigger. Okay. Once you are here, you will be able to see that everything is separated into several different sections. These sections are by data type or by application. You will also see some of the newer sections that have been added recently. And so these are some of the sections that I will be talking about, such as single cell analysis and others. So once you're here, each one of these sections represents a whole section of methods that are organized into a pipeline builder. And this pipeline builder is connected to some of the training that we offer on our OmicsLogic portal. So when you go through the training, you'll be able to use these tools in a practical way. When you use them for research, you will be given much more access to data storage and management capabilities. And then also we offer other types of use cases for this platform. So the basic idea of each one of these sections is to offer a pipeline builder. And this pipeline builder can be found as a demo based on a previously analyzed data set or a project, a demo project. So the way you would go into a demo is you would click on this dropdown, and then in one of these, you will find a preloaded data set with a prepared step-by-step pipeline builder tutorial that results in an understanding of inputs and outputs of each one of the steps. But the platform is really built to facilitate a streamlined research process organized around these different types of omics data, where in each type of omics data, you would get the output that is already structured and prepared for downstream analysis, so that later on, you can use it to visualize, analyze, and integrate some of the data sets. And so, as you can see here, you can both learn as well as translate that understanding into an actual research project. Now, what this does in terms of students that want to get into bioinformatics, when people mention bioinformatics, means so many different things, from very basic things like learning how to search properly on NCBI databases, to a little bit more advanced things like maybe learning how to analyze multiple sequence alignment data on short sequences, and all the way to big data. And so that is really where what I'm going to be sharing today is geared towards, right? How these kinds of tools can help simplify processing and analysis and integration of large data sets. Now, in each one of these sections, you will find that they are organized by logical steps or logical meaningful methods organized into groups that mean something, right? And so the logic is explained in several different ways. You can see it in pop-ups. You can also see that each step is color-coded. And because it is color-coded, you tend to remember exactly what each step means. And finally, at the end, it produces a reproducible workflow. So either where you stay on the platform and you use the platform, you remember what steps you took, or when you want to develop your own pipeline, you understand which method falls under which group, and you can design your pipeline in a logical way. In addition to that, we also offer different types of updated visualization options that we have recently been working to update. And those include standard visualizations like JBrowse 2, different types of interactive visualizations like you can see the PCA on Plotly, as well as custom things that we develop for specific projects that seek to integrate complex data sets together and provide a researcher with an opportunity to explore their data without having to deal with the complexities of multiple sources of information in several different formats. Now, when we think of this as a way to design a research experience, we can really think about as an application of some of these. And yes, Sarah, you can access the codes or scripts behind these pipelines. They are provided in the output. So if you have a research account, some of the methods that are written in R, for example, you can directly download the scripts at the end. Or what we do in the training is we actually go from these logical pipelines into the actual methods that are used and kind of go step by step into the things that you can do to extract this kind of information. So that's what I will be talking about today. So the way to do this is really to think about bioinformatics as a component of the project, of the biological question that is being asked. So, in many cases, when we think of methods like variant calling or maybe, non-metric dimensionality scaling, we can learn about these methods, but unless we see how they are applied in the context of a real biological question to answer that question, it is difficult for us to appreciate why the technical details play such a major role in what we are trying to do. And so to design an experience for those who are learning bioinformatics to appreciate some of the technical detail and to be motivated to learn how to use these methods appropriately, we have designed some of these tutorials around different types of projects. As you can see here, cancer, biotechnology, infectious diseases, and others. And so what I wanted to do today is briefly speak about 3 applications in metagenomics, single-cell Rna-Seq, and COVID surveillance, because some of these have really been seeing a major interest from a community of different researchers. And so I want to talk about some of the challenges and opportunities in these types of applications so that you can appreciate kind of how and why it's important to learn some of these tools and methods. So single-cell Rna-Seq, I'll start with this, is an area that has gone from kind of a very basic research application where primarily people were interested to understand different cell types by annotation, to more and more of a practical application where we are now even seeing multi-omics integration at the single cell. But importantly, there's more integration also with phenotype, for example, spatial Rna-Seq. And the basis of being able to use some of these kinds of advanced methods to really drive translational research is really understanding the logic behind some of the ways that we process such data. And we are able to address some of the technical challenges with the type of data that we have. So let's first talk about processing single-cell RNA-seq data, which tends to be a fairly automated process, but at the same time, it is a critical step that helps us address a lot of the technical variation at the beginning of this process so that our data is more reliable when we do the downstream annotation and visualization. So one of the major challenges is multiple types of sequencing data. I mean, 10x genomics has been recently kind of become the standard, but there are still many other different methods as well. And one of the challenges is here to address the artifacts. And so what we have done on the platform is we have prepared a number of tools that one can use to be able to process different types of data with different types of comparable methods. And as you know, in bioinformatics, a lot of the challenges are being addressed by looking at simultaneously running several different pipelines and comparing the results. And so here, this is really the approach to make sure that the data has passed the quality control, has been addressed at the batch and artifact level. And then also we have a very specific output that we can evaluate for how useful this is to do the downstream analysis. So this is challenge number one. And for anyone that has run these pipelines, you know that just putting together a single pipeline can take significant time. Running multiple pipelines to compare them in parallel is something that really becomes a huge challenge. And so having an interface that has a consistency and a reproducibility to it really allows you to run through that step of QC, prepare your data in a reliable way, and know that it is something that you can reproduce yourself. The downstream analysis also is a challenge because when we have these tables prepared, the matrices, we now want to analyze the data by comparing, for example, different clusters, different groups of samples to each other, and really know that our outputs, such as differential gene expression outputs or clustering annotation, is a reliable outcome. And so here we've designed a pipeline around SILRAT and producing the integrated matrix so that we can produce clustering and downstream analysis. And the important part here is that we have, as you probably all know, seen many researchers struggle with some of the basic challenges of SILRAT and these standard processing tools. For example, in anchoring, right? Knowing how to normalize and harmonize your data with Harmony, with anchoring and SILRAT, with other tools like Monocle, how to really validate some of the cluster annotation. There are many different tools. Do you go about it manually? Do you take some existing package from R? So those are some of the questions that really could be addressed in using such a pipeline. Now, to the point that Sarah was asking, how can you go from running a pipeline like this to actually being able to understand what goes into every single step and how do you use it? In our training, we have a training program called bulk and single cell Rna-Seq. And in that training, that's exactly what we do. We go through the pipeline, we take some projects, we see the inputs and outputs, and then we go into the coding part for those that are interested to really reproduce each step using R and trying to understand what are the specific details that go into every step that you need to master and kind of learn. So we provide you with a GitHub notebook. That notebook you can rerun, you can change the input and the output of the data, of course, with the limitations of the huge amounts of data that are out there, many of which are too large to process in some of these online environments. On the environment on your computer. And so you have to rely on the cloud to really kind of effectively run some of these pipelines. But that's exactly the objective. And obviously understanding how these objectives fit into the specific projects is a critical step, because not only do you understand the technical component, but you can apply it to address a specific research question and see how people that specialize in this domain can really be able to do that. And as a result of this training and developing these pipelines, we've now worked with multiple researchers at Louisiana State University and across the LBRN network, Georgetown University and UCSF, to support many of these research studies that are trying to integrate the more advanced methods like spatial transcriptomics and integrate single-cell RNA-seq data with bulk RNA-seq, for example. So really, if you have any specific questions about single-cell, different methods, different types of projects, I do encourage you to reach out to us. Our team here on the call can really schedule a time to discuss with you how your specific challenge or your interest could be addressed using some of these tools. Now, let me briefly switch gears and kind of talk about some of the developments and interesting new opportunities with microbiome research and data. And before I jump into that, I do want to leave just a couple of minutes. If you have any questions, please put your question into the chat box. I'll be able to see that and address it before I kind of run on into some of these other topics that I have in my slides. So metagenomics and microbiome is a field that has both a lot of challenges and a lot of opportunities. I think the challenges are really illustrated in the industry by two major factors. Number one, everyone's probably by now has heard of uBiome. a failed company that was kind of popularizing microbiome testing, direct-to-consumer testing, promising insights. Many other companies now have taken up that idea and promise to offer some insight into what's happening in your gut or what's happening on your skin. And you can really see that the benefits, direct benefits of such testing are limited. There's really such variation across individuals that it's difficult to find some very specific actionable insights. And in addition to that, I think the trust in metagenomic sequencing and such microbiome research has also been kind of questioned by the ability to kind of offer those tests at a clinical level. Not clear what exactly that clinical indication means. And so there's a lot of questions, I think, on what can microbiome can really tell us, practically speaking, about the human microbiome, but also how do the methods of analysis and the sequencing methods allow us to ask those specific questions in greater detail and get actual actionable outcomes and results. So to understand that, I think it's worthwhile to kind of go back and see, where did the metagenomics field kind of develop from? How did it develop into different types of sequencing methods? And where does it stand today? I mean, what could the real opportunity be at the end of the day to leverage what is available in the public domain, at least? So one of the things that, historically has been a big challenge is denoising, right? So one of the problems with metagenomics data, especially amplicon targeted metagenomics data, is that the data itself comes from millions and millions of microbes, and we have to very accurately sequence enough data from each individual microorganism to be able to annotate them using a database. And so at every stage of what I just described, there's a challenge. Number one, there's a challenge with having enough genomic data to sequence it accurately at a high enough depth. Number two, it is a challenge to amplify long enough reads to really know the sequence that will give us a high confidence in our annotation. Number three, the databases for annotation are really lacking some information at the lower taxonomy levels. And number four, at the end of that, we really do not understand in full detail how to interpret the differences in composition, be it differences in taxonomic abundance or differences in composition in terms of diversity and things like that. So to address this challenge, several different pipelines have been implemented on the platform, and I will talk briefly today about how those could be used. So really, we have three different approaches to metagenomic analysis on the platform, and I think they illustrate some of the challenges and how they're being addressed in this whole field. So these three include the whole genome metagenomic analysis, where you are sequencing long portions of genomes, and one of your goals is to kind of be able to piece together longer sequences beyond amplicon. And then that provides you with an opportunity to have more confidence in your actual annotation. Number two, KIME and Data2 for amplicon really focus on denoising that data, removing the inaccurate reads, and really focusing on the reads that only match their pairs, and then to annotate them with high quality databases, where you can use standard databases like Silva, which are available online for free, or proprietary databases that might have some specific focus and have some specific annotation. Now, in the context of such methods, I will also briefly talk about where this whole field is going beyond the processed data. So I'll skip this slide actually, but the point of this slide is that once you have that data processed and prepared, you are able to understand the scripts that go into each one of those steps, specifically the downstream analysis, where you can find such methods like PhyloSeq, NMDS, and others that provide you with these nice outputs. Okay, is it possible to modify these pipelines if required as per the user need? Yes, our team is constantly working with multiple researchers to provide them with new pipelines. Just recently, we've implemented a pipeline for single cell for different types of, for example, spatial single cell. All of these new pipelines come from research collaborations. So if you have a research project in mind, we can develop some kind of a collaboration. The result most likely is going to be that we will have a new section on the platform dedicated to that topic, where our team will review multiple methods, provide them as a user-friendly interface, but also use them to actually analyze the data. Now, in the microbiome space, I'll kind of go back to my topic. One of the major challenges has been developing a reference that is comprehensive enough and specialized enough around a particular topic. So, and Alessa, I'll address your comment here in a second. One of the original kind of objectives of these large-scale projects was to collect a baseline. And so we see projects like the Human Microbiome Project, British Gut Project, American Gut Project, Earth Microbiome Project, and others. Really, their goal was to establish the baseline of diversity. And so this could be compared to something like the HapMap project in the genomics space, where we try to understand, what kind of normal variation exists out there. Now, obviously, the challenge there is the number. And so I think the Human Microbiome Project is somewhere around 30,000 individuals. Really, when you think about the diversity of the microbiome, these are not large numbers. And so one of the challenges, of course, that the diversity is much greater than the sample size that we have. And so even for the baseline, it's difficult to establish that in a comprehensive way. But as a result of that, now some of those projects have become more specialized. And so here you can see cancer prevention study for the gut microbiome, Alzheimer's microbiome project, nurses' health study among nurses and others. And so, as you can see from baseline and kind of general diversity, now these projects are becoming more specialized. As a result, we now have a number of indicators that have been adopted for different types of practical applications when we think of individual microbiome, health, lifestyle, food habits, etc. And researchers have started really thinking about the microbiome and its establishment over time, where we can see a specific microbiome enterotype, for example. After that is established, how is it affected by the development and adult microbiome over time, primarily around food and lifestyle. And I'm talking here about preclinical or kind of healthy type microbiome. Now, as a result, we also see some sensitivities and preclinical conditions that are a perfect opportunity for early intervention and being able to design, non-clinical interventions like dieting, fasting, and other things, prevention of specific lifestyles. So that has become a huge interest because, as we all know, treating a chronic condition or an acute condition is much more expensive than trying to prevent it or manage it at the preclinical stage. So when we are thinking about microbiome and the opportunities, let's go and look at some of the things that we can find in these publicly available databases and think about how can that inform, some of the specific questions that we might design or we can think of. And here, this is an example of an extraction from the Human Microbiome Project, just the metadata of that project. And what you can see, obviously not in a comprehensive way, but you can see here that let's say I have a question, is there a microbiome difference between people who exercise rarely or regularly? Now think again, I have 30,000 people available on the Human Microbiome Project. As a result of that question, we were able to find only about 20 people from 30,000 individuals that have the same sex, the same age, the same habits, more or less, but only vary by their tendency to exercise rarely or regularly, right? So that is really the challenge. When you think of a question, always you need to go back and see what kind of reference do I really have. Nevertheless, some of those conditions could really be explored in a more detailed way. And as I mentioned, some of them have been established. So really the opportunity, and I think the challenge as well, is finding the right data or collecting the right data to inform your analysis and your hypothesis, right? And where you can start with the Human Microbiome Project, which is available for free. Now, another interesting application of the microbiome project is, of course, not only the gut microbiome, but also microbiome from other body sites. And one of those interesting ones is the skin microbiome. So in fact, a lot of people are interested in trying to differentiate between the effects of let's say, factors that we can control as opposed to factors that are more like genetics. And I think the microbiome sits kind of in between, right? So the microbes on the skin or any surface or gut are becoming established very strongly, even after depletion with, let's say, antibiotics. A lot of the same type of, composition comes back. And one of the examples here is an analysis of a paper and some publicly available data on the comparison between the acne and perioral dermatitis, another very interesting application. Obviously, in the clinical sense, this is becoming a more and more important component of how we think not only of the diversity and the types of microorganisms that are found, but also, as we know, they play a major role in the development of immune responses, intermediate immune responses, and metabolism. And so because of their, because of their role in metabolism, treatment becomes a natural next question. How does the metabolism for whatever enters our body affect the actual treatment efficacy? And here you can see a study published in Science in 2016, so really not a recent study, that already asked the question, could we find microbial signatures of treatment response? And I think this really has a very direct and strong implication for our ability to incorporate microbiome sequencing and microbiome biomarkers into the actual treatment development, which is a huge industry and a huge challenge as well. So now, again, if you have any specific questions about metagenomics and microbiome, our team has been working with a number of interesting projects at the basic research level, as well as the translational level, where we seek to integrate multiple sources of information and correlate them with microbiome signatures. And we are going to be making some announcements, I hope, in the near future about some applications that allow to do that, to really make that kind of service available to individuals. So if you have an interest in microbiome biomarkers, and metagenomic sequencing, I recommend, again, reaching out to our team and also seeing some of the upcoming opportunities to learn about and really talk to us more about your research ideas. Okay, so briefly, I'll take a look at the chat. All right, so excellent. Thank you for participating. About the specifics of metagenomic sequencing, I recommend we have a discussion. I think there are some really interesting developments in the metagenomic space. So LS, please reach out to us with that. And the question about cancer genomics, we do definitely have projects on cancer genomics. We will have a whole session dedicated to cancer genomics in our upcoming symposium. I recommend you join those. I won't be speaking today just because of the time limitations. Okay, so now let me speak briefly about Covet sequencing, some of the emerging questions in Covet sequencing and surveillance, as well as some of the methods that allow us to ask very specific questions about the evolution that is driving differentiation in strains, and obviously is of major concern as the public is more kind of comfortable, I think, with COVID being around, and the question is, where do we go from here? Where is this whole genomic surveillance and understanding of different clades going? So just so that we're all on the same page, let's review the basic idea behind genomic surveillance of COVID-19, and then we'll address some of these emerging new interesting questions that help us understand kind of the benefit of continuous surveillance of COVID-19. So at the basic level, right, there is a large genomic variation that helps us understand evolution and adaptation between different hosts, and maybe between different variation in the host, like immunosuppressed and other types of people. So the basic idea is that we have some level of variation. If we take a sample size from the genomic data, we will see that the amount of variation changes by sample, whether we take it at a different stage, or if we look at different portions of the genome. And the idea of that is coming from, a virus or a group of viruses infecting an individual, infecting a cell, as a process of replication inside the cell and evasion of immune responses at the cellular and the system level. The way that the virus can respond is by adaptation and changing its genomic sequence, right? So as a result, if we take that sample and we sequence it, we will see that we will find many genomes, and these genomes can be similar to each other or different from each other, right? And that is called genomic diversity. Or, in the case of COVID-19, we call these variants, right? So in genomics, traditionally, like cancer genomics or germline genomics, we talk about variants as individual SNPs or individual positions that are variants. In terms of viruses and COVID-19, we call those whole genomes, we call them variants. So those variants now represent some genomic diversity that we can start asking questions about. And so one simple question that we can ask is, what kinds of variants or what kinds of positions do we find that we can characterize, right? And so answering that question, identifying specific positions, either in FASTQ files where we have deep sequencing, or in assembled genomes, can help us, for example, categorize different genomes according to strain or according to their evolutionary divergence. That kind of analysis is helpful because we can try to associate clades of genomic diversity or groups that are genomically similar to each other with some phenotype. For example, a phenotype associated with the location or type of people, or the exact kind of phenotypic events that we can monitor clinically. Now, in addition to that, we now know that the kind of high-level genomic analysis of such data also can help us determine questions like, where did it come from? What species did it come from? Or how did it evolve from a particular location to the next location, right? So there's also a dynamic composition to it where we have the phylogeny and the time component, and together that is called phylogenomics. As a result of relying on phylogenomics and further expanding the phylogenomics to analyze the data that has been collected, important insights are primarily around the relationship between the genomic sequence and the way that this genomic sequence affects the actual protein. So because so much effort and research has been put into the characterization of the specific proteins that the virus produces, we can now understand mutability or the divergence, genomic divergence, in the context of proteins. And these proteins, of course, interact, right? The virus proteins interact with the host proteins. And because they interact with the host proteins, we can now take a look at crystallized structures of each individual protein, and we can model what actually happens inside the cell. And as we go through the training in separate programs that we have, like the COVID-19 training, we go through all of these steps. How do we go from sequencing data to phylogenetic analysis to then interpretation in the context of interaction between different protein structures at the host virus interaction level? Now, interestingly, that is just part of the story, right? as we continue thinking about virus transmission or where this whole thing originated from, we need to put in all of the pieces together to really appreciate what could drive such diversity in the future. Because ultimately, when we understand the different pieces and how they are connected, we now can think about intervention, but we also can think about the different processes that affect the continuous evolution. And as a result, I will speak about some papers that actually speak about how this is relevant for the future, post-vaccine and post, the pandemic kind of spreading, right? It's already everywhere. Where does it go from here? So as you know, the previous focus of such analysis was on different clades. And those clades have increased in number over time. The number of mutations seem to stabilize at some point, at the delta maybe point, and then started expanding again, right? And so one kind of practical outcome that we take from that, and we actually have a whole workshop coming out about that, we can actually just learn how to process the data and find the specific positions, variants of concern, or something like that that we want to report on. And that is the focus of that workshop. But in addition to that, I want to point to a paper that recently came out that talks about, and this is the paper right here, it's called Tracking Cryptic SARS-CoV-2 Lineages Detected in New York City Wastewater. This is an interesting article, and I'll share the link here for anyone who's interested to kind of go through that, that really leverages wastewater sequencing, which is sequencing of, again, a collection of different genomes in the wastewater, amplifying them with a specific amplicon sequencing approach, and then looking for novel mutations that are emerging. And the interesting conclusion that they arrive at is that they, first of all, identify specific mutations that are not found in clinical samples. Number two, they actually took those mutations and they checked in terms of binding and transmission and replication, how do they replicate in different types of cells? And they offer several hypotheses on how these could be driven by non-human animal reservoir, right? So what does that actually mean? That means that, because we live with pets, because we have rodents, because we have all of these animals around us, right? We are really starting to see that the virus, the ability of the virus, potentially it came from an animal reservoir, but potentially it continues to interact with these animal reservoirs, driving evolution, how it adapts and how new mutations come out, and of course we are now interacting with them again, right? So this is, I think, a very interesting study that shows that continuous genomics surveillance with sub-consensus analysis of data to look for new mutations that are not yet annotated, not just running through pangolin, but looking in depth at deep sequencing data, can help us understand how this virus is going to continue to evolve. So I think what I did in the short time that I had today is I was able to, again, go through these three topics as just a starting point and an introduction really to some of the things that we wanted to discuss. These three things were single-cell Rna-Seq, metagenomics and microbiome, and COVID surveillance. I know that there's a lot of training and opportunities to learn more about each one of these topics coming up. Specifically, I want to highlight a big event that we're planning called the Omics Research Symposium, where we have experts from all of these fields, from the fields of cancer genomics and precision medicine, from COVID surveillance, as well as microbiome analysis, and importantly, bioinformatics and data science. How can you learn? How can you apply some of these tools, and how you can drive translational research that actually reaches individuals, patients, and populations and makes a difference. And that's really what this workshop is all about. So again, thank you for letting me share some of these slides. I'll pass this back on to Mohit to kind of talk about some of the upcoming opportunities.
Thank you, Ilya, for this informative session. I think it was very nicely put together, and many of the participants are asking for the video because I think they would like to revise, and that is all right. We will be sharing the video recording as well. Now, about the less intensive part of the session, which is to discuss about the upcoming opportunities in the context of training, as well as like having these sort of interactive is this sort of overview session along with hands-on session and the interactive session that combines with that combines with asynchronous training as well. So it's a blended training that we are going to be doing throughout the next month. So the topics that Ilya was describing, so we have them spaced out one after another. So we are going to start with COVID research, like genomic surveillance of SARS-CoV-2 variant of concern and its reporting using R-script and the T-bioinfo platform analysis. And then we will be, after that, the next week, we will be doing NGS single cell data analysis and single cell RNA projects. That would be about demonstrating and looking at some of the projects that are there, as well as some of the research opportunities that one could think of through the publicly available data sets. And that would be followed by machine learning for omics data analysis, which starts the next week. So March 15th, Tuesday to Thursday, March 17th, where we will be going through about that how machine learning is applied and into these different studies and applications where we are using these high dimensional data to analyze and get insights from. So we will be using different techniques here, and and also to learn that how we can apply these techniques into big data sets, and also to utilize some of the tools from Python and R. So it will be also about coding, and as well as learning about the transition of data and how to extract meaningful information. That will be followed by the question that we answered about cancer genomics, and where we will be covering a detailed analysis using the T-bioinfo platform to analyze cancer mutations using this genomic data. And to sum it up, like it will be to discuss about also like the training and also the research and what we are doing with our OmicsLogic curriculums. We will be discussing that in our faculty and mentor workshop, where that will be an opportunity for faculty especially to integrate some of these curriculums such as introduction to bioinformatics, data science, and machine learning into their teaching or into the projects. So and obviously we discussed about the OmicsLogic Research Symposium that would be towards the end of March, where we will have guests from almost all around the world. It's a biannual symposium that we conduct. It will involve about all the outcomes that come from different types of analysis that we go through from different training programs, as well as research internships and research fellowships, as well as research projects that we are doing. So about the dates and all the registrations, I'll share with you. So if you can see my screen, we have the registrations available, which is shared by our team here in the chat for genomic surveillance. And then we have the SARS-CoV, then we have the single cell, and that would be followed by machine learning, and then the other programs. Along with that, we have some expert sessions coming up, which would be from our Omics Club. That would be also to address some of the important questions. So today we also discussed about structure biology. So how do we integrate structure biology from sequence to structure to function. So we'll be discussing that in our next event, which is on 25th February, 7:30 p.m. So you can also register for this if you're interested to learn about structure biology and its application in research project designing. So in our other in the workshop that we were discussing all about today, which is the genomic surveillance of SARS-CoV-2 and variant of concern reporting. So here we will have sessions starting from 1st March. So we will start from the very basic understanding of about data collection. So how this data is collected, what is the technology behind this data for those principal investigators, or for those research scientists who are starting with their career, or who are thinking about these projects or grants, can think about research ideas, starting from data collection, starting from utilizing, understanding about the known case studies. So this is something that we are planning to do in our workshop, starting from an expert session. So in this expert session, we will cover those aspects. Followed by that, we will be moving to more of a technical part, where we will be analyzing data in respect of a research project. So we'll be utilizing the bioinfo platform, where we'll be using specific algorithms and databases and data sets to analyze viral genomes, so that everyone gets an overview. And obviously, these sessions are also designed based on the level of expertise that is required for anyone to get or learn about these. So these introductory sessions on day 1 will be beginner level sessions, and followed by an asynchronous session, which is on day 2. But the third session on day 1, so we've, after having both the sessions at 8 a.m. and 9 a.m., we will have the third session at 10 a.m. CST, where we will be starting to discuss about the nature of emerging variants, many of those which we discussed in today's webinar, but in much more depth and in the context with the data set that we are analyzing at that point of time using the bioinfo platform. So after doing all of those sessions on day 1, as we are also like exploring as a participants, you will be also exploring, many of those who join these workshops will be exploring. So we have prepared a lot of resources for you to utilize, and those resources are something that you will have an opportunity to cover on day 2, which is to go through and to do these hands-on using the bioinfo platform, using the code playground, that all of those that will be described in day 1, and which will give you an idea to how to go through all of this through our online support. So this is the plan for the day 2, where you will be able to learn this from a basic to an advanced level based on your own level of expertise. So followed by that, day 3 is more research oriented, where we will be actually working on these data sets to look at different types of is it from clinical experiments sequencing data, environmental sequencing data. Here we will be discussing about the case studies that we have already worked on, and also come up with all of these different reporting systems and also different outcomes that we have found. So from these different analysis. So as we have already discussed this, so we will be moving from one session to another that actually is related to one another, but also at the same time, these type of data analysis could be independently done. And if you don't have time for the entire day, you can join one of these sessions and interact with us during the workshop, as well as get a recording for the entire workshop once it is over, and you can practice it on your own for 1 month, or henceforth those recordings are always available. So that's the so it will followed by the last session that we have on March 3rd, which will be to generate the report and utilizing R and looking at and analyzing that and relating it with the research question. So that would be the SARS-CoV-2 plan and the infectious disease. Those who are interested in infectious disease research and especially in SARS-CoV-2 could join that workshop with us and learn from some of our experts from industry as well as from academia. Followed by that, we have a Omics session. It's an free data science and student club and bioinformatics student club that I invite many of you to join, because here it's a rich community of students who are organizing these events for their own colleges, for their own universities, helping other students to learn about data science, OmicsLogic resources, and bioinformatics tools and techniques and courses that are available through OmicsLogic. So followed by that, as I said, we have on 8th to 10th March the single cell analysis, single cell transcriptomics data analysis workshops. So here we will be learning about some of the important pathways usually. So usually what we do is that we start with a broad overview of single cell transcriptomics, data analysis, and the applications. So looking from a perspective of where this is to and where this could be applied to. There are a lot of questions that many faculty and many students ask, which is to understand the actual application and if it is related, if it could be integrated into their research idea. So that overview is something that we start with, and we also discuss about different ways to get to work on single cell. So it could be through training of a student, it could be through research service, or it could be through collaborations. So that's what we are discussing, starting with the day one. And that would be followed by a project example or hands-on that would where we will be discussing about the downstream analysis that we discussed with Dr. Raghavendran, who is one of our co-mentors and is here in today's meeting as well. So again, the format helps you learn about all of this. So whatever you learn on day one, you get to learn all of this through available video recordings. So along with this, we will be providing you expert recorded video recording on day 2, where we will be also providing you the courses and tutorials that we covered, and the hands-on assignments on T-Bioinfo platform that gives you like a real experience of completing some of the things that we discussed and that could be relevant to your research, or that's something that you could be hoping to do maybe in the upcoming days in the future, or as a postdoctoral fellow, or as a faculty. So these are some of the So this is the plan for day 2. And after that, we have day 3, where we have an expert talk to summarize and understand where we are with single cell transcriptomics and its application in clinic. Followed by that, we will be moving to machine learning and data science. So how do we, after understanding the data from pre-processing data to cleaning the data, now we are trying to integrating and analyzing the data using machine learning methods. And in this short workshop, which is from 15th of March to 17th of March, we will cover important topics which are actually applied in many projects that we do and many student projects that comes across. So many of our research fellows who will be submitting their posters and who will be submitting their articles in journals will be benefiting from these sessions, where they could learn about dimensionality reduction and clustering techniques that is used for describing many of those research analysis results that we use in our poster presentation, in our reports, in our presentations, and as well as in the preprints and publications. So that's the focus of the workshop, is to first go about it, different types of data types that we discussed using the TBioInfo platform, and then apply machine learning techniques and explore the data, and to look at the dimensionality of the data and all of what we can understand from both exploratory analysis and then learning from the online materials that we have prepared for, especially for data science and machine learning workshop. So this is more about coding. So here we have R and Python, where you will be able to replicate some of the things that we practice from the GUI, as well as do downstream analysis, and especially when we are talking about obtaining great graphical results using using different types of methods, such as using predictive analysis as well as classification. So first we will learn about first we will learn about dimensional reduction and clustering, and then we are learning about predictive analysis. And in this entire workshop, you'll be also getting these tutorials like R for Omics bioinformatics. Here you will learn about different sessions that we take and go through these different analysis and steps. So the last session is also to get you through R and Python, and to get you through and to learn about how to do PCAs and how to use them how to build those using R and Python. And then it would be about the classification. That would be the last session on March 17th, to learn about supervised machine learning, and then to have a broad good overview about how we apply machine learning for next generation sequencing data, and what kind of graphical inputs that we can get to get a good research outcome from these sort of analysis, or to get some deeper understanding from this analysis. And followed by that, March 22nd, we have the cancer mutation genomic session, where we will be looking at the tools for genomic data analysis to interrogate genomic variations for cancer studies, and to perform and to look at the somatic and germline variant detection, driver and passenger mutations, copy number variation, as well as subclonal deconvolution. So this is something that is integrated in the TBioInfo platform, where I think many who are working on oncology, who are working on cancer genomics, or are interested in working in different types of, because a lot of data is available on public repositories, which is widely used by many scientists. So followed by that, the last session, the last workshop before the symposium, is on curriculum development. So we have our university partners whom we are working with, and where we have these curriculums where these curriculums are integrated for students to go through these specific trainings, like learning about different types of omics technologies, as well as integrating them into the curriculum, so that the student learn about the project-based learning, as well as the basics are covered through their curriculum within the university. So we already have many of these curriculums that we would want to showcase, and we would like to discuss with many of you who are interested. So that is the plan for March, and we are hoping that many of you could make it to some of these sessions and join us to learn with us, and also to let us know about what do you feel about it, and how can you be a part of many of these research projects that we are doing, which are based through this project-based learning, through these exploration of data which is available on public repositories, as well as the data that many of you are generating and could be supported through our expertise, through our tools that we discussed today, and through our collaborations that we are putting up together by getting all of us together and getting us to a platform where we can discuss science and actually also do science by using all of the resources that we discussed today. So that's what I wanted to tell you in my part of the presentation. But let me ask my team to share some of these links where you'll find all this information about the events, and also if you are interested in reaching out to us for any sort of research question, any sort of training related question or career guidance, then please feel free to reach out to us through email or filling up some of these forms that are available in these links that are being shared. So we have the research symposium workshops listed as Ojasvi has shared, and also the research symposium. So many of you could join the research symposium and showcase your poster and get an opportunity to get real feedback from some of the domain experts who will be joining this symposium. So as this is the page that you can see on my screen. So you can register. It's a free event. And I'm hoping that many of you who would have benefited from today's session would encourage others also. And please feel free to share this link with your contacts so that many of the students or interested faculty can benefit from these talks from these experts from industry, scientists, research scientists, and also specialization experts. So we will have collaborators from a lot of universities talking about their programs, and as well as industry experts from pharmaceuticals and biotechnology and agriculture and henceforth. So we'll be looking forward for your participation, and we'll be looking forward to see you in March for our events. It was great meeting you all, and I'll surely be looking if there are any questions that I can help answer. And if you're a faculty, yes, many of these workshops are available for free, and most of them are for free. Few of them are as they are training and you required specific T-bioinfo access, then there is a registration fee for the certification and T-bioinfo access. But most of the sessions are free for you to join and learn from, especially the research symposium. So again, thank you all for joining today's session. It was great meeting to you all, and it was great to see that many of you asked actually many questions. I'm hoping for you to ask more questions in the next time then we meet, and hope to see you, many of you, in the workshop where we will have more opportunity to discuss one-on-one.